Well, good morning, everybody. Being clumsy today. Well, happy Wednesday. Well, the DLW run finally finished last night after I left, so I will endeavor to do my best to get those data processed for you today and then get the spreadsheet sent out to everybody. Actually, I'll post it on Canvas when I get done, and I'll send out an announcement that it's available. I'll also try to get finished today. I've got an invert Zoe exam that I'm a week and a half out on, and I really need to finish grading today as well. So hopefully I can get all that done, but I'm gonna do my best to get the DLW water processed today. It means running it through. I have several little spreadsheet gymnastics that I need to do with it once I pull it off. But I've done it a lot of times, and so hopefully I can do that pretty quickly. The run went really, really well. The isotope analyzer had no issues whatsoever. There was only one sample that I got my water out of, so there ought to be data for pretty much everything. That doesn't mean that the isotope enrichments are all going to be great, but there's nothing I can do about that. So, but I'll have more on that later. So once I actually do all of the calculations and transformations on the data. So hopefully we can look forward to that tonight. Don't forget, if you are planning to retake the exam on Friday, if you have not talked to me yet, then please do so. So I will know how many of you there are going to be. I'm still trying to figure out exactly how I'm going to work that. And the reason I've got to think about that is because, with the lack of the webcam option on Respondus, that kind of changes how I need to approach exams. So I'm still giving some consideration to that. And part of that, at least for this, will be dictated by how many of you are going to do it. So please let me know if that's something that you want to do. And we should have a chat about it first, because you may be thinking that you want to do it, and I may actually talk you out of investing the time in that if I don't think there's enough benefit. For the groups doing surgeries tomorrow, if you can kind of give me a heads up as to what you're looking at for a schedule tomorrow, that I would be grateful for that. Beyond that, I think that's all I've got. Do you guys have any questions? No? Too early to think of questions. Huh? All righty, well, let's go ahead and get started. Oops, that's not what I wanted to do. That's what I wanted to do. Let's see. Come on. Thank you. All righty. Time to dig into the the peripheral nervous system and all of its components. So the peripheral nervous system is made up of 31 distinct pairs of nerves that are divided into actually 5 different regions, 4 primary regions. So of those 31, we have 8 that are in the cervical region up here. We have 12 that are in the thoracic region right here. You can see the division there. We have 5 that are in the lumbar region, 5 in the sacral region. Then there's this one coccygeal nerve that's sitting down there all by itself, which is, I guess, sort of the 5th region. We don't talk about that one very much. These tracts are split between the autonomic and somatic aspects of the peripheral nervous system. So you've got one set of nerves that are servicing the autonomic nervous system and the other set that are servicing the somatic arm of the peripheral nervous system. Come on, there we go. Okay. So here is kind of the typical way in which one of these spinal nerves works. So the afferent side is the dorsal root of these spinal nerves here that will provide, they're going to connect to a peripheral sensor of some sort, and it's going to provide input into the CNS. And then the dorsal root of the nerve is the efferent side of it, and it will transmit via motor neuron to either a muscle or some sort of gland, depending on whether we're talking about somatic or autonomic response here. Okay. So each of these nerves, and the way you got to think of these nerves is more as nerve tracts rather than single nerves. And I think I've mentioned this before, you know, when you look at something like the vagus, which you will all play with a lot during your rabbit surgeries, the vagus nerve actually contains a whole bunch of nerves that basically are both afferent and efferent. So it's not just a single nerve; it's a single nerve tract, and these spinal nerves are exactly that way as well. Okay, and you'll see some evidence of that as we move forward today. Okay, I think I already said that. So half of these nerves are connected. You know, the dorsal half is afferent, and the ventral half is efferent. Is going to go to various sorts of effectors, again, which could be muscles, glands, or whatever. Okay, I mentioned that these nerves are split between the somatic and autonomic divisions here. There are some slight structural differences between the spinal nerves that service the somatic versus those that serve the autonomic. The autonomic arm of the peripheral nervous system, the efferent side will always go through what we call an autonomic ganglia down here. And the reason for this is that the output is then from the autonomic ganglion going to be directed to some very specific places in which will drive the efferent response to the autonomic signal. On the somatic side, efferent is pretty much just going to a skeletal muscle someplace, and so it's a little bit simpler. The structure of the nerves themselves are a little bit different in that, and I'll actually have a bullet point, I think, about this later. But on the somatic side, the axons of the nerves are heavily myelinated, whereas that's not the case on the autonomic side. On the autonomic side, they tend to be either lightly myelinated or not myelinated at all. Okay, okay, and I think we've talked about that already. Okay, yeah, we've talked about that already. Okay, so the autonomic nervous system largely exists to control things like blood vessels. And when I talk about controlling blood vessels, I'm talking about things like vasoconstriction, vasodilation. It's going to control your heart. And actually the control of the heart by the autonomic nervous system is actually somewhat complex. And we'll get into that, not just a little bit later today, but when we get into the cardiovascular system. And then, of course, various aspects of your viscera, your gut, your glands. It is related a lot to the secretion of hormones. So it's really integrated into the endocrine system. In fact, when we look at the autonomic nervous system overall, it's really integrated into a lot of things. It's really hard to think of the autonomic nervous system as being this discrete unit, physiological unit, because it integrates with so many different things. So when the next topic, when we actually start formally talking about the cardiovascular system, you're going to see the fingerprints of the autonomic nervous system all over that. Okay. When we get to your oral reports and we start talking about the endocrine system, you're going to see the fingerprints of the autonomic nervous system all over that. So this is the one challenge of teaching physiology from a systems perspective, in that everything is just so integrated that it's really, the lines between one and another are really great. And a lot of this, this compartmentalization that we want to do, again, is more to satisfy our human need to have things nicely broken up into neat little packages, because it's easier for us to deal with. All right, so how more specifically is the ANS laid out? So they generally receive signals from some very specific locations. The autonomic nervous system is divided up into two primary units that we looked at last time, the parasympathetic and the sympathetic nervous systems. The parasympathetic nervous system is the nerves from there are going to arise from the craniosacral region of the CNS. So from way up here and way down here. Okay, this is where the parasympathetic nervous system is going to come out of the spinal nerves. The sympathetic nervous system is everything in between that. So it is the thoracolumbar region. So all the nerves out of here will service the sympathetic system. The sympathetic and parasympathetic systems typically are going to have kind of opposite effects on the target organs they serve. And both of these are going to service the same target organs for the most part. There are some exceptions to that. So the sympathetic system typically excites or arouses a target organ. And as you might guess, the parasympathetic system typically calms or relaxes a target organ. So when you think about sympathetic and parasympathetic nervous systems, you're thinking about the maintenance of homeostasis. Okay, one tugging, tugging to excite, one tugging to relax, and wherever that balance is your homeostatic state. All right, it's involved in all kinds of regulation. So if your body is trying to respond to some sort of an external stimuli or a series of external stimuli, or respond to your need during exercise, that's going to be a total autonomic response. And you're going to shift between sympathetic and parasympathetic control depending on what the body's needs are. Okay, so it is a constant bit of tug of war. So the efferent pathway of the autonomic nervous system is typically going to involve a it's always going to involve a preganglionic nerve. First, let me go back and mention about the somatic again, just to state that it's a much more simpler design than the autonomic. So you basically have an efferent nerve that's going to have this heavily myelinated axon that's going to go to a skeletal muscle. But in the ANS, you're going to have a preganglionic nerve that's going to interface with an autonomic ganglia right here. Okay. The autonomic ganglia then is going to direct the efferent signal to whatever target organ it needs to go to. Okay. So the ganglion, remember, ganglia are a location of local processing. So it's going to kind of relay it wherever it needs to go. And it will go there via a postganglionic nerves. The characteristics of these preganglionic nerves and these postganglionic nerves can be actually quite different. And in the case of the autonomic nervous system, the postganglionic nerves will connect to smooth muscle, cardiac muscle, glands, the digestive tract, a whole bunch of different things that are internal. So Preganglionic nerves, regardless of whether we are talking about the sympathetic or parasympathetic arms of the autonomic nervous system, are cholinergic, which means what? What does it mean for a nerve to be cholinergic?
Uses acetylcholine.
That's right. So it's going to release acetylcholine, okay? Which means that the autonomic ganglia that it is connecting with is going to have acetylcholine receptors on them. And these acetylcholine receptors are very specific, and we'll talk about those in a moment. The postganglionic nerves of the parasympathetic system are also cholinergic. So they also are going to release acetylcholine as their neurotransmitter. There are a few cases where parasympathetic nerves will release nitric oxide. Anybody know what nitric oxide does? Well, I'm going to tell you. Nitric oxide is a powerful vasoconstrictor. So, or I should say, maybe more correctly, it is a powerful smooth muscle contractor. And it may not make sense to you, based on what we've already said this morning, that a parasympathetic nerve would initiate a smooth muscle contraction, but there are cases where it does, and we'll point those out in a little bit, and why that's still kind of relaxing. The postganglionic nerves of the sympathetic system are all adrenergic, which means what? You could probably look at the slide. Yeah.
They release norepinephrine and epinephrine.
Yeah, they release norepinephrine. And actually, as far as the nerves go, they release norepinephrine primarily. Epinephrine is absolutely part of the sympathetic system, but epinephrine is released primarily from the adrenal gland and is delivered most often as a hormone. There are some exceptions to that. There are a few neurons in the CNS that will release epinephrine. There are a few neurons in the ANS that will release epinephrine, but the vast majority of, or the primary release point of epinephrine is going to be from the adrenal gland. All right. So, I mentioned this already, but preganglionic nerves of the ANS are lightly myelinated. Postganglionic nerves are unmyelinated in the ANS. Now, some things that I want to point out here. So here we have the sympathetic, some examples of sympathetic arrangement up here, parasympathetic down here. So we mentioned that all preganglionic nerves are cholinergic, but the receptors on the autonomic ganglia here are all nicotinic. Okay, so just the same sort of receptor you would find on skeletal muscle. The receptor in the adrenal gland that results in the release of epinephrine. They're all nicotinic. Okay, so, and that's going to be the same is true even for the parasympathetic. All preganglionic nerves are going to release acetylcholine, and the acetylcholine is going to bind to a nicotinic receptor. The sympathetic system, the postganglionic nerves in this arrangement here and this arrangement right here, okay, these are all adrenergic, And so the target organs are going to have some sort of an adrenergic receptor. The big point that I want to make here is generally these receptors fall into one of two categories. They're either alpha receptors or beta receptors. The reason why this is important is because often these two receptors can elicit opposite responses. So remember how we were talking about the fact that neurotransmitters can elicit different types of responses from a postsynaptic cell, depending on what type of receptor the cell had. Well, this is an example of that. You know, if it binds to an alpha receptor, it may do one thing. If it binds to a beta receptor, it may do another thing. And when we get through in a couple of slides, we'll show you a chart and give you some examples of that. The other thing that's important to remember about these receptors is that the binding affinity of these receptors can be different. The reason why this is important is that you're going to be doing surgeries where you're going to be giving differential, you're going to be giving injections of something like epinephrine in low and high concentration. And so I want you to keep in mind that the affinity of epinephrine to an alpha versus a beta receptor could be different. And I'm going to let you go out and figure all that out because you're all smart people. And so when you deliver different concentrations, you may be highlighting one type of receptor versus the other if they are both located in the same place. So for example, if you are targeting a blood vessel, one receptor may trigger vasoconstriction, the other may trigger vasodilation, and which one wins out may depend on the concentration of the neurotransmitter that's there, or the hormone that's there in the case of epinephrine. Does that make sense? If you've got a high concentration of something, then it's conceivable that even though a receptor has a lower affinity, if it's there in, say, higher density, it may dominate the effect. But in a lower concentration, that might not be true. They might selectively bind to a receptor that has a higher affinity. When we look at the parasympathetic system, the postganglionic nerves again are cholinergic, but the receptors on the effector organ are all muscarinic. So remember how we talked about, you eat those bad mushrooms and you go on parasympathetic overload. This is why, because the effector organs all have muscarinic receptors on them. Okay. And we already said that somatic motor neurons are heavily myelinated because you're trying to get a muscular response and you want it to happen really fast. This just illustrates much of what we saw in the last slide, except now we have one of these common diagrams that we see in just about every, not just physiology book, but even general biology book, that shows many of the connections that are made by both the sympathetic and parasympathetic system. It doesn't show them all, but it shows many of them, just so you can see how wide-ranging the impact of the autonomic nervous system is. And when you look at like everything below the neck, you'll find that the vagus nerve is integrated with most of these things. Okay. And over here, we're just illustrating pretty much exactly the same thing that we illustrated on the last slide. It's a little more detailed. You can see down here in terms of the release of epinephrine that it actually highlights the bloodstream here, illustrating that epinephrine is delivered in the bloodstream, making it a hormone, because by definition, that's how hormones are delivered in the bloodstream. If you've got a biological messenger that's delivered in the bloodstream, it is technically considered a hormone. Okay, here is a table that illustrates a lot of the functions of both the parasympathetic and sympathetic nervous system here. And I like this table a lot because not only does it tell you what each of these systems specifically do, but it also identifies for the sympathetic system the particular type of receptor that's binding to. Okay, so we can look at things like, we can jump here to the heart. So we talk about the heart, we'll talk about the SA node. The SA node with the sinoatrial node is the pacemaker of the heart. It is what generates the signal, the contractile signals for the heart. And you can see here that the sympathetic system is going to bind to a beta-1 receptor right here, which means it will increase heart rate. But conversely, the parasympathetic system will decrease heart rate. If you had no autonomic control of heart rate, your heart rate would be around 100 beats per minute. Yet in a healthy individual, it's actually closer to 70. In a trained athlete, I've known some whose heart rates are in the 30s, okay? Which tells you that in healthy individuals, the heart rate is largely under autonomic control or parasympathetic control, all right? But the autonomic nervous system impacts the heart in more ways than just rate. You can, in terms of the atria, okay? The strength of contraction in the atria is controlled by the sympathetic and parasympathetic system. And this is through two types of beta receptors. And then again, the parasympathetic decreases the contractile strength. The AV node, which is the interface between the atria and the ventricles in the heart. So the signal first causes atrial contraction. It's received by the AV node. It delays there a little bit so that your atria and ventricles don't contract at the same time. And then it transmits it, okay? That is also influenced by the autonomic nervous system. So the sympathetic is going to increase the conduction velocity. This corresponds with increasing heart rate, again, through beta-1 and beta-2 receptors. The parasympathetic system slows down. So the very last thing in the cardiovascular surgery that we will do with a rabbit is we will give it a high dose of ACH. And the reason it's the last thing we do is because it simulates AV block. And so at that point, you can actually stop the heart of the rabbit. So it's the last thing we do, okay, before we euthanize, okay? And so, again, that tug of war between the sympathetic and parasympathetic. The ventricles. The sympathetic system is actually strongly integrated into the ventricles to increase contractile strength. The parasympathetic, not so much. It can have a little bit of an impact on the strength of ventricular contraction, actually to reduce it, but doesn't strongly affect that aspect of heart function. We'll go down here and look at arterioles here. And the main thing that I want you to look, you'll note that the parasympathetic doesn't do a whole lot in terms of vasoconstriction, vasodilation. But what you see here, depending on whether the sympathetic is interacting with alpha or beta receptors here, determines whether it's going to cause a constriction or a dilation. So we can see, for example, oh goodness, let's see here. Let's find. Actually, let's go down here to the lungs for a second. You'll note here that in the lungs, the sympathetic interacts with a beta-2. I think it's beta-2 that beta-2 receptor, and it causes the bronchial smooth muscles to relax. Does that make sense for the sympathetic system if it's something that should excite? Yes, no, maybe so. Does it make, yeah, go ahead.
Yes, because if you're under stressful situations and your sympathetic nervous system is like taking charge, like you want more oxygen, so.
Exactly, that is exactly correct. So if you think of something like fight or flight, okay, the last thing that you would want to do if you needed to up and run is to constrict your bronchioles. And so in this particular case, it causes relaxation, increases the diameter so airflow is easier so that you can up your aerobic metabolism. So in essence, it is a proper response for excitation of physiological performance. Similarly, when you look at the digestive system, the parasympathetic system promotes peristalsis. The sympathetic system shuts it off. It will eliminate peristalsis largely because, go back to this concept of fight or flight, if you're trying to get away from someplace, the last thing you want to do is devote any of your blood supply to digestion. That's all going to your muscles, and so there is no point in spending energy contracting your intestinal smooth muscles. So sometimes sympathetic and parasympathetic will promote a response that on the surface may seem the opposite of what those particular arms of the ANS are designed to do, but when you put them in context, they make sense. Does that make sense? Yeah.
Does this process like have anything to do with why chronic stress can have effects on the body?
Can you be more specific?
Like effects on the heart or like have there any digestive issues from chronic stress as well?
I'm not certain. I do know that if, when you are under chronic conditions, it can relate to some issues that aren't necessarily directly related to the autonomic nervous system, but could be driven by the autonomic nervous system. So for example, if you have high blood pressure, if you have chronic high blood pressure, your arteries, your arterial walls will start to get hard. And so that could put you more at risk for things like stroke. That's a byproduct of that. So you could be having a prolonged sympathetic response that will lead to that, but the sympathetic system is not actually doing that, if that makes sense. It's a byproduct of that. Other questions? Yeah.
Why are the alpha and beta receptors only on the sympathetic? Like, is it just the pattern, the kind of receptors found in that system?
I'm sorry, I didn't.
Why are the alpha and beta receptors only on the sympathetic side?
Because they're adrenergic receptors. They won't bind to acetylcholine.
So that's
Acetylcholine there actually are, even within nicotinic and muscarinic receptors, there are actually subtypes of those as well. Those are just specific types of receptors that will bind to norepinephrine or epinephrine. So remember when we talked about fast versus slow chemical transmission, we talked about these adrenergic receptors are going to be binding to signaling systems. So they're not ion channels, where all the acetylcholine receptors, they're fast chemical transmitters, they're all going to bind to ligand-gated ion channels. Did that answer your question? Anything else? This just gives you some more examples of what the sympathetic and parasympathetic system do, and I'll let you sort through these. These are in, I think these are in the Widmer text. I think that's where this comes from. Okay, just to get a little comparative here for a moment. all, organisms of all levels will show some type of autonomic organization in their nervous systems. Here's an example of from frogs where they exhibit sympathetic control over cold tolerance. And they looked at this in two species, leopard frogs and bullfrogs. And what they showed here was they have a basically a measure of sensitivity to cold on the y-axis. And then in this case here, frequency of just direct nerve state stimulation or concentration of various sympathetic messengers here, in this case epinephrine and norepinephrine. And basically what you see is that as the level of sympathetic input increases, you get a higher level of cold tolerance, illustrating that, that there is sympathetic control of cold tolerance. So if these frogs get into an excessively cold environment, they're going to get increased sympathetic stimulation to help them deal with that. Okay, that would be considered an excitatory response. All right. But, it's probably not surprising that we would see something like that in frogs. They are a vertebrate. They're ectothermic, but they're a vertebrate. But Vern Heinrich, who is a really, really well-known old-time physiologist, demonstrated autonomic-like control in a sphinx moth, an invertebrate. This is really a cool experiment. So what we have here is we have body temperature on this axis, right here. We have time on the x-axis here. And then body temperature was measured at three, I guess, or four different spots here along the body. A thoracic measurement up here and 3 abdominal measurements down here. And then as temperature was increasing, they measured heart rate and the intensity of heart contraction. And these are labeled A, B, C, and D, and you can see up here the points at which they were measured. All right. And so what Heinrich determined is that as temperature increased, the heart worked harder to circulate blood within the sphinx moth, in part to control or stabilize the thoracic temperature. Okay, thorax right there. All right. And determined that this control of heart rate was driven by a nerve that ran between the thorax and the abdomen, an autonomic-like nerve in a sphinx moth. Then he did experiments where he found that if you cut this nerve, then that regulation of heart rate and contraction intensity disappeared. And the temperatures got out of control, and the thoracic temperatures would rise above 50 degrees when they were heated pretty substantially. And 53 degrees was the same temperature that a dead moth would get to. So that served as his control in this experiment. So in this case, even a sphinx moth has some very low-level type of autonomic control in this case. So this is characteristic of a wide diversity of organisms. Okay, so organisms exist, and we've talked about this pretty extensively. There is a great diversity in the complexity of nervous systems depending on the needs of whatever organism you're looking at. But there are certain generalizations about nervous systems that are true of all organisms, regardless of what you're looking at. So there are 3 main generalizations. First, neural networks are made up of specific connections between neurons, and each set of connections serves a specific function. sends specific types of information to the CNS, which in turn elicits specific responses. In other words, these networks are designed to do some very specific things. The connections are established during development. So as you are going through the entirety of your ontogeny, you are building and perfecting these connections. They are maintained throughout an organism's life, and in some cases modified to meet specific needs of the organism. This would be particularly true in the CNS. And this is an interesting one. If some aspect of a neural network falls into disuse, you could lose that function. In other words, if the body isn't using it, it gets rid of it because why energetically support it? So an example of this is in the military. Periodically, you'll get soldiers that are in battle that will end up with some sort of a paralysis that's just driven by the brain. It's a psychological issue. And one of the first things that they try to do is get that soldier moving, get them using the limbs. Usually it's the legs. Because if you don't use something long enough, you actually permanently lose the use. Even in individuals that suffer an injury-based paralysis, they will sometimes go through stimulations of nerve tracts just to keep them active. Presumably in the case that something will pop up at some point where a treatment that doesn't exist now may exist down the road for them. Just trying to keep all of that stuff kind of remembering what it's supposed to do. So the last thing that you want to do is to just stop using something because your body may just discard it if that is the case. Questions about this? All right, generalization number 2. The synaptic, metabolic, and electrical properties of individual neurons determines the way which each neuron responds to the sum total of the synaptic signals impinging upon it. Okay? The synaptic, metabolic, and electrical properties of individual neurons determines the way in which each neuron responds to the sum total of synaptic signals impinging on it. So basically, if you're thinking about this, we talked about how all the characteristics of neurotransmitters, okay? The particular types of receptors that will be part of neuronal structure will dictate how that neuron is going to respond to a signal. It's got a very specific type of structure, and that structure is designed for it to respond a very particular way. Okay. So in this particular generation deals with the response of individual neurons. And then generalization 3, the complexity and variety of function is directly related to complexity and variety of neural circuits present, not the variety of signals. Okay, so you build, and this actually in some ways relates to the first generalization, because you're going to build a neuronal circuit to do some very specific things. The neurons in that neuronal circuit are going to be constructed to do some very, very specific things. The level of complexity in terms of those specific things it's designed to do is going to be related to the complexity of the structure of that neuronal circuit. The classic example of this, I think, is when you look at the difference in complexity of the brain of the diversity of organisms that we have on this planet. Clearly, we as humans have the capacity to do more complex things than a flatworm. At least I hope. And that's because we have neuronal circuits that are far more complex because our neural processing needs are far more complex. You can even think about this, if you want to think about this in terms of like a circuit board. Why do our computers continually get better? Because we build more complex processors that are capable of doing more things. It's the structure of those processors that allow for better performance of more modern computers. Our brains are very much the same way. Does that make sense? This here is probably the most important generalization. It certainly best explains why some organisms have better capacity to perform than others. All right, questions? All right, two types of neural networks, and we actually covered both of these. We talked briefly about both of these in one of our very first slides in this topic. Okay. The first is a sensory filter. That is the input side to the CNS. All right. Sensory filters are designed to bring in complex signals, get rid of those things that you're not interested in paying attention to, and focusing on the things that you are interested in paying attention to. So this type of a network, or at least component of a network, filters out unwanted information. And the visual system is a classic example of this. And so here's an example of a frog looking at a background who is hungry and interested in foraging. When we look at page number 1 here, we see a frog staring at an image where there is a bug that is moving, the background is staying stationary, the plants are dark, then the background of the plants is light. And so they are measuring output from 2 nerve fibers which are associated with recognition of food in some way. And they just call them type 1 fiber and type 2 fiber, both are firing here. Now below this, they show the frog 3 different alternatives with regard to images. The number 2 here is a stationary image, but is an inverse of the image above. Note that type 1 fibers do not fire, type 2 fibers do, suggesting that type 2s are more generally associated with the visual system, but not associated, say, in the search for food. In example 3 here, we have a case where it's the same image as example 1, but the whole image is moving in this case, not just the bug. Again, type 1 does not fire, type 2 does. And then finally in 4, they just have a black bar here and move the whole image. Same thing, type 1 does not fire, type 2 does. So there's something special that the visual system is picking up on when the only thing moving in the image is the bug. This is an example of a sensory filter. Okay, I'm out of time, so I will stop there. Any last minute questions? Okay, we should finish up this topic on Friday and actually dive into the cardiovascular system. What?
Yeah.
Is there a reason to not have class Friday?
It's a holiday.
Is it a holiday?
Yeah.
Oh goodness, you guys are more in touch with that than I am. So, oh goodness, I had scheduled an exam for Friday, didn't I? You don't have school, okay. That may mean I have to push it back to Monday, huh? Well, I tell you what, those of you who are planning to retake the exam, if you want to take it on Friday, it's fine. If you want to take it, if you prefer to put it off till Monday, I can do that. I did. What holiday is it? What am I missing? Oh, that's why I don't get it. Okay. No, believe me, I was oblivious to that, so thank you. Okay, so give me a few moments. Yeah, I think I have I did see your email. I'm pretty sure I have enough anesthetic. That's gonna be the determining factor as to whether you can do it now or not. But I'm pretty sure I have enough anesthetic, because you need to, before you euthanize them, you need to anesthetize them. That's just the best way to do it. And then I have to get you the euthanasia drug, so. But give me a minute, I gotta get out of here for Jim, so.
If you do have it early this afternoon.
Oh, you want to do it this afternoon? Oh, that's fine. So I have faculty senate. And what time is early this afternoon? Oh, I have class at 1, so you'll need to do it either before or after that.
Okay.
Okay. So I have class till 2. So,
Sounds good.
okay.
They may be able to do it at 2. I'll check it with them. And are you gonna be around your office before class and stuff?
Yeah. I'm here. I will be here. I gotta get moving here. So I will be here until probably maybe as late as 3 o'clock. I have a faculty senate meeting at 3:30. I'm still trying to decide whether I'm going to go do it in person or whether I'm going to do it via Zoom, because